Okay. Okay. Hi, everyone, and welcome. Thank you so much for joining us today. My name is Marta Campabadal, and I'm the Senior Manager at the Open Academy. As you registered, today's session is all about communication in science, specifically how researchers in both academia and industry share their results. We'll look at different ways they communicate and explore what makes good or bad communication. And we'll also illustrate the consequences of poor communication. Our speaker today is Professor Annemieke Aarsma-Roos from the Department of Human Genetics at the Leiden University Medical Center. She's also a longtime member of the Open Academy Schools faculty, where she leads the ethics breakout exercise and the session on translational research. So some of you may know her already. Before we begin, just a quick note. We'll be using Slido during the session for polls. So please keep an eye out for instructions on how to participate, and Amike will explain it to you a little bit later. Have your smartphone ready. And if you have questions during the presentation, you can write them in the chat. And I think with that, I'm happy to hand things over to Annemieke. And whenever you are ready, Annemieke, you can start sharing your slides.
Okay, thank you very much, Marta, and I'll share the slides. And if things don't work, then I count on Marta and Gemma
Yep.
to intervene. Um, so let's first make this smaller. Okay, so welcome, and today we'll talk about communication in science, and my disclosures don't really have anything to do with that, but for completeness sake and for transparency, I show them here anyway. Feel free to take a screenshot if you're interested, but we're not going to talk about this today. So we're going to talk about communication in science, and then, of course, primarily communication in science about therapy development. And I'm sure you're all aware how therapy development happens. Usually you first start in cell models, then you move to animal models, then you move to clinical trials to test the therapies. And of course, the next step is always more important. So you start in cells just to see, does this work, yes or no? And then you start in animal models, does this work in a living organism with organs and circulation? And then does this work and is it still safe in humans? So each step is important, but the next step is always more complex. And that means that success early on is no guarantee for success in later steps. And so if you read a scientific paper where they test things only in cells and they say now a treatment is around the horizon, that's not really true, because there's a lot of next steps, and the answer of the next step can be, well, actually this works in cell, but it will never work in mice or dogs or humans. Also, a clinical trial is, of course, an experiment in humans. So a clinical trial is not a therapy. It's a test of a potential therapy. And then what we will say as researcher and as patient is the trial failed. And what we then mean is the trial showed that the therapy that we were testing didn't work. Well, if the trial showed that the therapy didn't work, then the trial was successful, because the test was, does this therapy work, yes or no? So a failed trial is a trial where at the end, the result is inconclusive. So we don't know whether the treatment worked or not. So I think with this introduction already you see that there is many ways in which communication can go wrong when we talk about research and therapy development. So now we'll go back to the basics to discuss a bit about how communication works, and then we'll go into the different aspects again. So communication happens between humans. And of course, there's a sender and there is a receiver. So we can say something, but it may be perceived differently by the person who hears it than we intended it. Because in our head, we have concepts. So whatever word we say, there will be concepts around it. The person that hears it, they also have this. So when we say a certain word, it may have a very different meaning implication to the person who hears it than to the person who says it. And very often you may have said this, well, I know I have at least said it. When you talk to someone and they don't understand you, you will say, "You're just not listening to me." Well, yes, they are listening. They just interpret what I say in a different way than I intended for them to hear. And I think this is where miscommunication goes very often wrong. And this is, of course, not just happening in science, this is happening in general communication as well. So now first we're going to do our first exercise to make this more clear. So if you have your mobile ready, you can screen the QR codes and then it should bring you to the Slido page. If that doesn't work, you can go to slido.com and then enter 3449766. If you don't have a mobile ready, or if for whatever reason you cannot use a mobile, you can also type it in the chat and then Marta and Gemma can tell me, or just shout it out yourself, then I can hear you as well. It's all fine. So I see people already seeing how it works. I asked, what do you think of when I say Barcelona? And we are already getting answers. We're getting Eurodis. Yeah, if you went to the summer school or the Open Academy, that is Barcelona. You think about that. People think about Spain, nice city, Mediterranean, Gaudi, Sagrada Familia, Paella, sun, football, Freddie Mercury. So you see a lot of different things that people think about. They're all right. Like nothing is wrong. They're all correct. But it's because of your personal experience with Barcelona, there's things that you first think of and think of secondly. I was once robbed in Barcelona, so I will also have that as a as a concept. But you will see like one word will trigger different things in different people just because of well, our personal experience. And maybe to illustrate this with another example, if I say Hamilton, some people will think of the founding father of the US, other people will think of the Formula One racer. And if I'm in New York and I say I'm going to see Hamilton, you will think, okay, she's going to see the musical, not the Formula One driver. If I'm going to a Formula One race, you will think she's probably not going to see the musical. So the context matters as well. And this is how communication works, but you can easily see how things can go wrong because I can say Barcelona and think of the Sagrada Familia, but if you think only about football, then the communication can go weirdly. So this is how communication works in general and also with scientists. But scientists, so how do we communicate about our research? We use publications. So we've done research, we've done our experiments, and then at some point we think, okay, we now have enough to share this with the world. We write it up. So this was the hypothesis. This is how we did the experiments. These are the results, and this is the conclusion we draw. Then we submit it to a paper, to a scientific journal, and our peers, so fellow researchers, will check. Does it make sense what they wrote, what they did? Did they use the right tests? Did they use the right models? Does it all make sense? And then sometimes they will say, well, like this one experiment that would be really good to do because then you can really draw your conclusion. So they will propose do an extra experiment. Or they will say, well, you use this control, but that is a really bad control. You should have used that control. Or they say, well, you draw this conclusion, but we think you actually should have drawn another conclusion. So you get feedback. And sometimes after one round of feedback, the publication is done, but sometimes it takes 2, 3, 4, 5 rounds of feedback before the publication is finally accepted and then published. So when you read a publication, usually this has happened. So there has been this peer review where fellow researchers have commented with the idea is constructive criticism. So then you can think, so at least there's been some review process before something is published. But of course, that doesn't mean that everything that is published is correct. Because there's two types of journals. There's journals of good standing, good reputation, where this process is done rigorously. But there's also now predatory journal, because how publications work is that if I publish, it's now common and mandated in many countries that you publish open access. So everyone can access the results and the research. And well, many funders say, well, if we fund your research, we don't want you to own the data. We want you to share the data. So everyone should have access. But this open access we pay for that. So my publications, I pay to the journal an amount of money to publish. And now there's some clever scientific journals that say, well, that's a good business model, because the more we publish open access, the more money we will get. And then they don't really care about the scientific quality. They just want to publish as much as possible. And researchers, how are we evaluated? Well, how much do we publish? So for us, publishing a lot is a good sign. Then we are evaluated better by our universities and by others when we apply for grants. And these journals say, well, if we publish more, we get more money. So this is their business model. And some scientists, if you do sloppy science and the good journals say, we're not going to publish this because you didn't do a good job, then these predatory journals are your way out. So that means that if something is published, it may still not be good. It may be very bad science, but somehow people manage to get it published because of predatory journals. If you want to see which journal is predatory, if you Google predatory journal, there will be lists that will appear where you can see, okay, is this journal that I'm now reading the publication from, is that a high reputation journal or not? So that is one way, and it used to be relatively good with the peer review, but now we have the predatory journal that make things, well, less credible. Then also what researchers will do, they will present at scientific conferences. And there will be a scientific audience, which is a very critical audience, because then normally when you publish, you will have 2, 3, 4 reviewers. Now you have a whole room full of reviewers that critically listen to what you have to say. So researchers are used to, when you present your data, to use several ways to show the evidence, to convince their audience. And sometimes they will forget that if they present to a room of families or patients, that is different, because, well, we don't need to convince you with 15 different ways. We can just show you once and then you will probably believe it. So that is, sometimes you may think, well, aren't they saying the same thing 15 times? Yes, that is because that's how we're used to talk to each other. And then we're not realizing that when we talk to other people, we have to do it in a different way. But this is how scientists communicate. But then usually when therapies are done, are tested in clinical trials, there's pharmaceutical companies involved. And pharmaceutical companies, they communicate in different ways. Because if they're public, they have to report to their investors. And when there's a clinical trial result, they have to report this to their investors. And they do this usually with a press release. So the results that are reported in the press releases, that is not primarily for the scientific community or for the patient community. That is for their investors. And this is how the system works. So this is not peer reviewed. So this is how the company has done their analysis, and these are the results. So there's no criticism yet from fellow researchers. And very often they will have a quote of an academic in there who then says, well, these results are really promising, and well, I'm very happy with these results. But they can pick who they select for that quote. So if they know I'm very negative about certain type of research, they're not going to ask me to give a quote, obviously. They're going to ask whoever is their friend to give a positive quote. So definitely look at it with a grain of salt, because this is how these press releases work. Then later, the results are also published in a scientific journal. And it's actually mandatory now. If you do a clinical trial, you have to publish the results. And well, not everyone does it, but it's almost all companies now adhere to this. And these publications are then reviewed by fellow researchers. And then you also get into the nitty-gritty of the data. The press release is just a summary, but now you can see, well, these were all the data. This is how the analysis was done. And it's peer reviewed, so they cannot bluff their way or fluff the data up, because fellow researchers will look at it and they will be critical. And usually also these publications involve academic authors, because it's the company that coordinates the trials, but of course the clinicians are involved in conducting the trials. And this takes time. So it takes time to write a publication, and then it has to go through several rounds of reviewing. So the press release usually will be out there, and then 1, 2, sometimes even 3 years later, you finally get the publication where you can dive in and see what was really happening in the clinical trial. Because this takes so long, very often these trial results are also presented at conferences, and then you get more information than you get from the press release. And usually companies will use academic presenters, but again, be aware that the slides are approved on by the companies. So again, if the company feels positive about it, they're going to invite a presenter who was involved who's also positive. So there's like, I'm not saying they're lying, but they have an influence on who to choose to present and how to show the data. So again, I mentioned already, you have this miscommunication where people think, well, you're not listening, you're not understanding what I'm saying. Scientists are humans, so we can make mistakes, we can be wrong. So we can have an hypothesis that turns out not to be true, but we also can misinterpret data, etc. We're also enthusiastic about science in general, and usually especially about our own science, and we're not trained to present to lay audience. And well, this I'm not very happy about this. I'm trying to sort this wherever I can with students to train them how to do this properly, but this is not something that is part of the normal curriculum. So usually when scientists present to the audience, that is lay or patients and family, they will use jargon, like words that mean something for researchers or clinicians, but that say to the common man are like, you don't understand. All these clinical terms for everything. Yeah, if you don't know what it means, then it's really difficult to follow what the presenter is saying. Also, we commonly overestimate how much the audience knows because we think, well, yeah, we all used to have biology, so people should know how these things works, forgetting that, well, yes, we had biology in high school, but then we've been working with biology for years and years and years continuously, while the average human being, they will only have had biology in high school, and that may be 10, 20, 30, 40 years ago. We can then not assume that they will remember all of that. Finally, scientists are trained to be critical, and they're very often critical about the work of other scientists, but they will have a blind spot to their own work. So they will be less critical there. And again, like scientists are human, this is a very human trait, but also be aware of that. Usually scientists will be less critical of their own work. So now we're going into some examples, as Marta already said. So what I do, so every day, every work day, I read a paper, a scientific paper, and then I summarize it on social media. So Blue Sky now. So if you're interested, feel free to follow me or to just have a look. And I try to summarize it in such a way that it's understandable, but sometimes it's hardcore science, and then of course it's more difficult. But what I also do, so if people make a mistake in my view, I will write it down and I will say, look, this doesn't make sense because of such and such and such, or if there's a misassumption. So due to reading this paper every day now for 5 years, I have some ideas of things where people often go wrong and common mistakes that I will use to illustrate, because I know this day and age, everyone has access to all the research. So you might look into the research, and sadly, even if you look into scientific literature, it's good to be critical. And I know you were not trained as researchers or scientists, most of you, but you still have to be critical. And I hope to give you some tools to see whether research was done properly or not, with some examples. I'm from the Duchenne field, so there mainly will be from the Duchenne field. However, they apply beyond Duchenne. So you can apply them also to your own disease. So first, I'm going to ask, the next slide will be a question again, but it will be about this graph. So what we see the diaphragm function. So the muscle that you need to breathe, we see the function. And the function, higher function is better. We see an MDX. MDX is a mouse model for Duchenne. So this mouse doesn't have dystrophin, and this mouse has muscle pathology. So diaphragm function is impaired. So it's, at the moment you can see in the graph, it's 5. Now we have a treatment, doesn't matter which one, in the MDX mouse, and we see that diaphragm function improves. So it goes from 5 to 8 in the treated mice. And now my question to you is, what does this mean? How well does this treatment work? Do you think it doesn't work at all? It works a little bit, it works quite well, it is amazing, or you don't know how well it works? And again, there's no right or wrong, and it's also anonymous, so don't worry about answering what you think. It's just to illustrate how these things work. No one thinks it's amazing, and no one thinks it doesn't work at all. But it seems to be, I don't know how well it works quite well, or it works a little bit. And I think, well, I would agree mostly with the people who say, I don't know how well it works. And that is because there was one important thing missing. And you very often see this in scientific papers. They will show the disease model, they will show the treated model, but what they will not show is the wild type, the healthy mouse. And what we see here, the healthy mouse has a diaphragm function of 100. And while in the previous graph, when the wild type was not there, the increase from 5 to 8 seemed to be, well, quite substantial. Now that we know that normal is 100, I think you will all agree, well, yeah, it does something, but it's really, it's a little bit. It's not much. And this is the thing, without knowing what is the healthy situation, it's really difficult to interpret a treatment effect. And now we had function where at least higher was better, but there's even papers where they show the disease model and the treated model, and then whatever change they say it's an improvement. And changes up are improvement in something, changes down in something else are improvements. You don't know that. If you don't know what the healthy situation is, well, if things go up, they might get worse or they might get better. So it's really important to check, did they include healthy controls, cells or mice? That is something that sadly is still quite commonly not done. What you also sometimes see is that researchers are a bit detached. Now, I'm a researcher, so I can say this. Researchers are sometimes a bit detached from the human reality, so from the patients. So some researchers never actually see patients. They will see their animal model, they will see their cell models, but they might never meet a patient. And that can lead to a sort of a very narrow view because they're so familiar with their model that they focus on their model. And sometimes these models, yeah, mice are not humans. So part of the pathology may be the same in the mouse and the human. That is great. And that's why we use the models. But sometimes there's pathological aspects that you only see in the mouse and not in the human. And then researchers can say, okay, this little aspect that we see, we're going to try and treat this. And then they see less of this. Well, let's just give an example. The mouse has a different skin coat than it normally would have. So normally it would be black and now it's gray. And then they do that therapeutic effect and the mouse returns back to its black skin color or fur color in case of mice. Well, if say the color of your hair, if that is not seen, that the patients have a different color of hair than before the disease started, then normalizing this is probably not going to have a therapeutic impact in patients. And I now make a really weird example, but we've also seen that the mice will, for instance, develop bones in their muscle in Duchenne. You sometimes see this. Well, that is not something that happens in the Duchenne patients. If you prevent this, well, great for the mouse model, but since it doesn't happen in Duchenne patients, there's not really a human translatable angle. So that is also, be careful. If you see that a study is done and they focus on a pathological aspect where you say, well, but that's not part of my disease, then maybe it's not the best therapeutic option. And then something I didn't introduce yet, but also academic institutions will have sometimes press releases. So when there's a scientific publication, especially it's in a high journal that's well received, like Nature or Science, where people say, well, that's really very high impact, really important if you publish there, then the academic institute will be proud and they will have a press release where they summarize the findings. And of course, these press releases are not written by the researcher, they're written by people working in the communication departments of their institutes. Of course, they will do a check whether what they say is correct, but they're, well, sometimes inclined to inflate the findings of the publication. And I'm going to show you now one example of how this can go wrong. So there was in 2018, so now 7 years ago, there was a scientific publication in Science, so one of the top journals. And what it boiled down to, they had a dog model for Duchenne, and they treated it with CRISPR, so gene editing. And only in one dog was this successful. So in one dog, the gene editing restored the production of a missing protein, and instead before treatment, the dog didn't produce dystrophin. After treatment, it produced a Becker type dystrophin. So a partially functional dystrophin, not normal, but better than nothing. So this was the publication. And in the publications, authors were very careful not to oversell. They said, look, so we know now in a dog model, so that's bigger than a mouse, in a dog model, we can do this, but all we do now is showing that this missing protein is restored. There's a lot that still has to be done. How long will this persist? What will the impact be on function of the muscle? It only worked in the muscle, not in the heart. So there were a lot of things that still had to be done. Then the press release was inflating it a little bit, and then, of course, the media picked it up. So first thing that came out was an online piece that CRISPR fixed muscular dystrophy in these dogs. Well, it didn't fix the disease. It restored the missing protein. But there was no study whatsoever on how this slowed down the disease. So bit of an oversell. At least this one says human trials are far off. Then a few hours later, things were expanding. Muscular dystrophy was halted. Again, this was not shown in the paper. Are humans next? Question marks. And then the next day, gene editing successfully stopped Duchenne muscular dystrophy in dogs. Human trials are next. So you see how quickly this escalates from a scientific publication that was really careful not to oversell things. This publication was in 2018. We're now in 2025. I know there's some CRISPR trials for Duchenne going on in China, but nowhere else are there any concrete plans for CRISPR trials in humans, including the people who wrote this publication in the dog. So this just shows you how quickly these things can escalate, and just don't believe all these headings that you see, because again, people have a tendency to inflate things because that is how clickbait works and how you can sell your message. Then something I want to discuss is drug repurposing, and I'm guessing you know, but just for context. So a drug may be approved for one condition, and then you can think, well, maybe this drug also works for my disease. Or so as a researcher, the disease I'm working on, as a patient or a patient organization, the disease you're focused on. Because, well, it targets this part of the pathology in that disease, and that pathology is also part of my disease. And of course, if something is approved for a disease already, there is a lot of data. So that should speed up things for your disease. So the idea, the hope is that things can then be developed quicker. But there's certain things to consider. And well, if you've been to the past open academies, we sometimes had a breakout session on drug repurposing as well. So then you may be familiar with some of these questions, but this is really good to ask yourself, because if a treatment is approved for an acute disease, so like an infection, most rare diseases will be genetic, so they will be chronic. So if a treatment can be used for a week or a month, that doesn't necessarily mean that you can also use it forever and ever. So that's one thing to consider. Then, if the treatment is approved only for adults, many rare diseases affect children. So is there any safety data in children? Because if there's not, that would be an extra thing to collect, and that will slow things down. Is the route of administration possible? In Duchenne, patients often have swallowing problems. Then if you have huge tablets, that's not going to work. And then sometimes what they do, they will show in animal models, like this approved drug for disease X or Y now also works in the MDX mouse, or whatever other model. And then if you check and you see the dose they're using, you see that they're using a much higher dose than what is used in the clinic and what was used in the mouse model to develop it for the first disease. So that high dose, you then check, would this be possible to give this to humans? And then you will see, no, this is a dose that will not be tolerable in humans. So then, of course, again, yes, it may work in the mouse. Mice are really resilient. They probably can cope with that very high dose, but humans cannot. So then what is the point? And finally, the fact that it's safe in one disease doesn't mean that it will also be safe in another disease, because there can be aspects that play a role there. Again, using Duchenne as an example, Duchenne patients continuously have muscle damage. So that means there's protein degradation, and the kidneys continuously have to cope with this protein degradation. And well, there will be protein in the urine, they will try to reabsorb it from the urine. So the kidneys have to work harder. And then something that may be safe in a regular human who has good kidney function might not be safe in Duchenne patients because they have this kidney problem. There's other diseases where there may be liver pathology. And in someone who has osteoporosis, for instance, that might not be a problem because their liver is completely healthy. But if part of the disease that you're now wanting to use it for is liver problems, then maybe it won't be safe. And finally, for many rare diseases, there's symptomatic treatment with medication, and then the repurposed drug might interact with that. So that's another thing to consider. So it's not as clear as, well, this can just be applied. Again, we're going to use an example. So I'll first introduce it and then ask the interactive question. So this is sildenafil, and you all know sildenafil because that is the generic name for the compound Viagra. So Viagra causes blood dilation or blood vessel dilation. And in Duchenne, we know that there is a problem with blood vessel dilation. So it's the blood vessels are not dilating when they should. So then someone thought, well, maybe sildenafil can improve things. So they tested in the MDX mouse model. They tested when already there was cardiac pathology, they started giving Viagra. And then they gave it to half the mouse and half the mouse they did not. The mice that they gave Viagra had reduced pathology after treatment. So there was less fibrosis, so scar tissue formation in the heart, and the heart function improved compared to when they started the treatment. While in the mice that didn't get the Viagra, they had more fibrosis and they had worse functioning. So then, of course, also this was picked up by the internet. But then, of course, the question is, Viagra is already approved. It's been on the market. It's been used by many individuals. Knowing this, that in the mouse model, Viagra can improve heart function, would, if you're a Duchenne patient or if you had a son with Duchenne, would you give it? If you are a clinician and you're listening, would you prescribe this to your Duchenne patients? Well, I see some people already say, well, I would first wait for the clinical trials. I would not do this. I might consider it, or yes, I would immediately go for this. And again, I'll give you some time to think and answer. And well, I think I like that most people think I would wait until the clinical trials are done. That would also be my cautious thing. But I can see why you would consider it. Like I would definitely not say say no, because the mice did better and it's approved. So after the results in the mice, two things happened. One, a clinical trial was initiated because we as a field thought this needs to be studied further and we need to test it in patients. In parallel, there were families, especially in the US, who said, well, we're not going to wait. We are going to either order Viagra online, or fathers pretend that they had erectile dysfunction, would get the Viagra prescription, and then they would give this to their son rather than take it themselves. So they were self-dosing, thinking Viagra is approved, so we might as well. Clinical trial was done. Of course, this took a few years. And the result was it doesn't improve the heart pathology in Duchenne and Becker patients. So what worked in the mouse didn't work in the human. And if you go into the paper in detail, what you see is that actually it makes the pathology worse in Duchenne patients. And I think this really highlights that these families that were treating themselves and thinking, well, what harm can it do? Well, it turns out humans are not mice. The mice heartbeat is 600 beats per minute. Ours is a lot less. It's a different physiology, it's a different anatomy. So the fact that something works in a mouse doesn't mean it will also work the same way in a patient. So I'm with you on waiting for the trial results. I can see why people would consider it, but I think this really shows it's better to first test things in trial before you dose yourself. Because you can think, well, what harm can it do? It actually did harm. And so as a preclinical research, of course, I work primarily with mice. And we very often get the comments that what we're doing is useless because we test things, it works in the mouse, and then a clinical trial is done and things don't work. So there's no predictability. And I think with the Viagra example, that was correct. It was tested in the mouse, and then it turned out in the humans it didn't work. But very often what we see is that people go too fast to their clinical trials. So what you do in mouse, you first do proof of concept. Does it work, yes or no? And then if things work, you need to optimize it. That is the more boring. Knowing whether something works or not, that is exciting as a researcher. But knowing which dose works best, which regimen works best, which route of administration to take, that is less exciting because you're optimizing and well, you don't get this thrill of does it work, yes or no. And these things take time. And then sometimes there's a tendency to say, well, this takes time, but with rare diseases, usually we're working with progressive diseases where we're spending time, patients don't have time because every day somewhere someone might lose a function. So then what the developers, the drug developers will say, we're not going to do all this extra preclinical work, we're immediately going into the clinical trial. And that is a risk because how do you then design your trial properly? What dose do you use? What regimen do you use? If you don't have this information from these extra studies, it's really difficult to design your trial properly. And what you get then is that you get these truly failed trials where the results are inconclusive, or even worse, where you conclude that the treatment is not safe or doesn't work just because you didn't use the optimal dose or regimen. And we've had this example in the Duchenne space a lot, where trials fail because of safety concerns. So the treatment is toxic. And then you say, yeah, but maybe a lower dose, would that have worked? Yeah, that might also have worked, but we never tested this in the mouse, so we just went with this dose. So now you have probably overdosed the patients, are in the area where it's not safe, and you conclude, well, we can't do this because it's not safe. Well, actually, a lower dose might have been equally effective and not have these side effects. That is, of course, not good. Another reason is the opposite, where you may conclude that something is not effective, but maybe it would have been affected if you used a higher dose or a different regimen. And I think the problem, so you may think that going quicker is better because the patients don't have time to lose. But if that then leads to a suboptimal trial design, you end up with trials that are inconclusive or tell you the wrong thing. In the neuromuscular field, we now have the TreatMD Advisory Committee for Therapeutics, and I actually returned from one such meeting yesterday. And this is a way that we do to de-risk treatment development. So both academic and pharmaceutical companies can apply for this advice. And it's a multidisciplinary panel. We have people with preclinical expertise, clinical expertise. We have, of course, patient representatives, people with regulatory expertise, people with manufacturing expertise, toxicology, etc. So everyone that you would need to properly develop your treatment and your trial design is in the room, and then they can look on all different angles to the applications and maybe say, well, you should really do this preclinical study because without that you can now not design your trial. Or you designed your trial, but your outcome measures were not chosen. So for instance, the patient representatives will say, well, you're now testing this, but I don't really care about this. These aspects are much more important to me for my activities of daily living. So it's a really multidisciplinary approach to try and de-risk treatment development. And I think it has benefited the field. And so it was set up for the neuromuscular field, but I know now the ataxia field and the glioblastoma field has adapted the same model. So then moving to scientific publications again, sadly, there's a bias to things that work. Scientific journals are more inclined to accept publications showing that something works than showing that something doesn't work. And that means that if 3 people test the same thing and one shows that it works and the other 2 shows that it doesn't work, it's possible that only the one that shows that this therapy that works, only that publication is published. And the other 2, the work is not published. So, and of course, if 2 independent people find the same thing, that makes the conclusion much stronger. But it's really difficult to also publish. Look, they showed that this chemical compound worked to alleviate the symptoms. We now confirm this. That is more difficult to publish. If you say, well, they showed that this works, and we tried the same thing with the same model and the same compound, we don't see that it works, that is incredibly difficult to publish. And this is really detrimental because in the Duchenne field, there's patient organizations that are willing to fund clinical trials. But then if they think that something works, well, 3 other groups try to make it work, but cannot, they will only see what's published. They will not see that, well, maybe this doesn't work as well. It's not very robust because 4 people tried and only 1 succeeded. So to alleviate this, the Duchenne Parent Project in the Netherlands has funded our group, but also some other groups around the world, to have a validation lab. So if someone wants patient organizations to publish something or to fund a clinical trial, and there is some questions about, well, how robust is the data, they can go to some of these validation labs and say, can you just repeat this preclinical study to see whether this works, yes or no? And if the answer is yes, then we feel more comfortable funding this clinical trial. And if the answer is no, then apparently we need to do more work. So that has improved things. And I think now what's now more and more common is that when you submit your work to a scientific journal, you also put it online in the bioarchive. It's not yet peer reviewed, so it's like work in progress, but it's there and people can see it. And I think that is definitely helping because then you don't have this problem that if you show that something doesn't work, people can still find the fact that it doesn't work. So this bias is happening for scientific publication, but it also happens in social media. So something may be tested in a clinical trial and something might even be approved. The people where it works very well in, these families, these parents, these patients are much more likely to show that on social media than the people that have no treatment effect or reduced treatment effect. So be aware of that. There is a bias in social media. You will only see the top 5% of people where whatever treatment works really well. Usually speaking, it will be working a lot less well in other people. So just be aware of that. I will now go over 2 final examples of how scientists can also fool themselves. So we have a tendency to so strongly believe in our own hypothesis that we apply that to our own work and ideas. And maybe I think in view of time, I will skip the first one and just give the second example because I think that is the most important one. Well, people can see the slides if they want afterwards, so that they can read the first one. So I will skip this in view of time and mainly go over this one. So this one is about a food supplement. And this is why I use it because I know many individuals and patients use food supplement. So beta glucan is a food supplement. It's available over the counter and it has anti-inflammatory properties and anti-fibrotic properties. part of the Duchenne pathology is inflammation and fibrosis. So, knowing this, would you suggest this compound to Duchenne patients if you have Duchenne or are a parent of Duchenne? Would you consider using it? Because, well, it should alleviate the pathology. So I would consider it, I would do it and use whatever other supplements I can get my hand on. I would like some more information or I would not consider this. And I think we see that the majority of people either want more information or would consider it, which I think is, well, makes sense. It sounds appealing. Well, first something about supplements, because the fact that you can get them without a prescription, you might think that they are safe, but there's some things to consider. There's usually a dose that they suggest where you can think, well, more is probably better. No, it's not, because these supplements can have impurities. So if you overdose yourself, so take more than the suggested dose, this can harm you. These impurities can harm you. Also, supplements are much less restricted than drugs. So there can be variation in batches. And supplements can have negative interactions with prescription treatment. So in case of Duchenne, steroids, cardiac medication, they can have a negative interaction with these. So if you consider taking a supplement, I would recommend discuss this with your clinician to see whether there is any of these negative interactions. If there's not, then your clinician will probably say, well, go for it. But don't think just because you can get it over the counter that it's safe and that it will not be harmful, because sometimes there might be very harmful interactions with treatments you are taking. So back to this beta-glucan study. So authors tested this in the MDX mouse. These mice don't make dystrophin because their mouse dystrophin gene has a mutation. They have pathology, inflammation and fibrosis, which the beta-glucan should improve. And this is an oral treatment, so they put it in the drinking water, the mice drank it, and then they got their beta-glucan. They used a wild-type control with dystrophin, and they also used a untreated MDX mouse. And what they found after the treatment, they report in the text, is increased dystrophin. This is the results. In the text, they say we see increased dystrophin. If you look at the results, it's not that much increase. So this is the healthy mice. This is untreated. Well, treated is slightly more. NS means not significant, but still they say there's an increase. And here, so this was in the blood. This is in the diaphragm muscle. Again, these are the levels in the wild type. Lower in the untreated mice, and then here treated. Again, not significant, but they said we see an increase. Now, knowing this, Duchenne patients don't make dystrophin. We now have this beta-glucan thing that increases dystrophin. Would you now consider it, taking it? Yes, I want more data. I have questions, or something weird is going on here. Well, at least no one is saying yes, but probably from the way I phrased my answers, you already had some idea that something strange is going on. And indeed, so beta-glucan should reduce inflammation. It should reduce fibrosis. The MDX mouse doesn't make dystrophin because its dystrophin gene is not working. So it cannot produce this protein. Having this supplement is not going to have any change to the gene. So why would this mouse suddenly now make dystrophin? It makes no sense whatsoever. Also, dystrophin is present in the muscle. It shouldn't be present in the plasma. So authors show it's increased in the diaphragm. It's also increased in the blood. The fact that it's in the blood doesn't make sense. So what is most likely is that authors are not looking at dystrophin. They're looking at something else. So they're looking at an artifact. And they think it's dystrophin, but it actually is not. And I think what I find surprising, so this was published in a scientific journal of good standing, but neither the author nor the peers, so the fellow researchers that reviewed it, none of them realized that it makes no sense that the treatment increases dystrophin. So no one said, look, this doesn't make any sense. And I think that is a bit disturbing, but it also tells you, like, we can have a system and we can have some fail safes, but still it's never perfect. So these weird things can still be published. So don't believe everything that is published. And then authors also make a claim, and they say, well, they went the wrong way around. So they first did a trial in Duchenne patients, and then afterwards they tested in mice whether it worked or not. Um, and then they said, well, in humans, we also already saw improvement in strength in treated patients. Well, I looked up the human study. Dark blue is before treatment, light blue is after treatment. On the left, ambulatory patients, on the right, non-ambulatory patients. They say there's an improvement. Well, I don't see an improvement. I see that the blue line on average is about the same as the dark blue line on both sides. So they say there's an, so their text says there's an improvement, but the data say there is not. And I don't think these individuals are actively lying. They're just so convinced that this works that whatever difference they see, no matter how minute it might be here and here, this is how they interpret it. So take home messages. Scientists are humans. They may be over enthusiastic, and that is why it's good that scientists do a reality check, what is still needed. But it's also good that when you read or listen to people who are scientists, that you do this reality check. Okay, so now what stage are we in? Are we in the cell stage, animal stage, clinical trial? Is this approved by the regulators? And because that will have an impact. So if you hear news about Duchenne or research in another field, is it a press release? Probably meant for investors. Is it a scientific publication? Higher credibility. It's a news report, lower credibility. Approval by the regulators, higher credibility. Even if it's published, you now know that you have to check whether there were controls in place and whether whether the rationale made sense. And then also a message that I give to researchers, but also to people working at patient organizations, is not to hype therapeutic development. Because when people decide whether to go into a trial or not, or whether to have an approved treatment or not, there's always a risk. And if the treatment effect is hyped, is oversold, people will have unrealistic expectations. And then the risks that they're willing to take, or the burden that they are willing to take from a clinical trial or a treatment, will be higher as well, because they expect a lot. Well, if they knew that the treatment is probably, well, it's going to do something, but a lot less than what they expect, then the burden of the trial, or the burden of the treatment, or the risk of the treatment, they might have a different assessment about that. So having too much hype really risks that families accept burdens that are too high. And sadly, in the Duchenne space, we now have had a few patients who died after getting gene therapy, where we know that the therapeutic effect of gene therapy so far has not been shown. And if anything, it will slow down pathology a little bit. So it's not a cure, but some families thought it was. They were willing to accept the realistic risk of having liver failure. And sadly, in a few patients, that is what now happened, with very serious consequences. So with that, I thank you for your attention, and also for answering all my questions. And of course, I'm also happy to answer questions that you may have for me.
Thank you, Annemieke. I see some great reactions from the participants. I don't see any questions in the chat, but maybe someone would like to unmute and share some thoughts or ask Annemieke a question before we wrap up.
And maybe to say, so the nice thing is that I will leave the Slido open. So if you didn't participate yet, you can. And for the people who watch this later, they can also still participate in the voting.
Fantastic. Okay, I see no questions. Thank you, Jenny and Sandra. They're posting on the chat, thanking you for the presentation. It was very interactive, and it's nice to have this format once in a while. Just as a reminder that on the 25th, we have another masterclass about practicing an elevator pitch with our colleague Nora from Eurodis. If you've registered, you must have received email from me with an assignment. And if you haven't registered, I'm going to put the link in the chat and just check it out and see if you're available and want to join us. Sue says that this was the best presentation she's ever had. It is amazing. Thank you everyone for joining us. I don't see any questions. I think we can finish here. Bon appetit. I'll leave you to grab something to eat, and I'll see you probably, for the next masterclass. All right.
Thank you. Bye.
Bye.
Thank
Thank you,
you.
everyone.
Goodbye.